Hi, my name is Priyal, and my topic is on chemoresistance in BRCA-mutated ovarian cancer. So a little bit about me. So as I said, my name is Priyal. I currently live in New Jersey, and I'm a junior in high school. So in the future, I want to pursue medicine. So I think Polygence was a great way for me to learn about the research process and a platform where I had guidance while learning something that I'm passionate about. So huge thanks and a shout out to my mentor, Elizabeth, through helping me through this process. So throughout this presentation, I'm just going to go over multiple aspects of chemoresistance. So first, just like the different methods that I use during my research, as well as the mechanisms of chemoresistance, different treatment implications, as well as prevention methods. So first, I'm going to be defining what ovarian cancer is. So ovarian cancer is the second most common malignancy in women over the age of 40, following only breast cancer. It's also the most common cause for gynecological cancer deaths. Ovarian cancer can be due to different causes such as hereditary or genetic factors and even carcinogens. So my project focused mainly on the genetic causes of ovarian cancer through family syndromes, specifically hereditary breast and ovarian cancer syndrome, also known as HBOC. So the genes that are affected within this syndrome are breast cancer 1, also known as BRCA1, and breast cancer 2, also known as BRCA2. So the BRCA genes are tumor suppressor genes, which have the function of regulating cell division to prevent the onset of cancer. So BRCA genes repair DNA mutations caused by errors in cell division, which can prohibit cell regulation and result in the uncontrolled growth or division of cells, also known as cancer. The incidence rate for women that have BRCA1 or 2 mutations within ovarian cancer populations is 12 to 14%. And although it seems like a relatively small number, women who possess BRCA1 and BRCA2 mutations have a 50% and 30% risk of developing ovarian cancer, respectively. So next, I'll be talking about chemoresistance. For patients who have cancer, medical professionals may use a variety of different treatment methods, the most common being chemotherapy, which is when cytotoxic chemicals are used to kill cancer cells. The cancer cells may develop resistance to chemotherapies through varying mechanisms, rendering these therapies as ineffectual. Chemoresistance is usually observed through cases of uncontrollable cancer or patient relapse. Around 70% of patients in an advanced stage of ovarian cancer recur within 2 years despite chemotherapy treatment. So how can we combat resistance? By understanding different mechanisms of resistance, doctors can offer alternative treatment options for patients. However, even despite efforts to develop alternate treatment procedures, additionally working towards chemoresistance prevention will help increase the effectiveness of primary treatment approaches. My research considers mechanisms of chemoresistance, such as DNA repair by cancer cells, drug efflux, and autophagic processes to review chemotherapeutic treatment intervention within BRCA-mutated ovarian cancer. So as per my methods, so there were 2 databases that I consulted. I consulted Google Scholar and PubMed, and then listed above are also some keywords that I used within my search. So now I'm going to be talking about the mechanisms of chemoresistance. The first one being DNA repair. So normal cells use DNA repair for restoration purposes. However, DNA repair becomes a hindrance within chemotherapy treatment because it repairs the damage that chemotherapy is purposefully inducing in cancer cells. So while BRCA-mutated cancer cells are originally developed through a lack of DNA repair for genetic mutations in somatic cells, which are cells that aren't gametes, chemoresistance emerges due to the ability of cancer cells to become tolerant to DNA damage through using alternate DNA repair pathways to repair DNA lesions. Platinum chemotherapeutic agents such as cisplatin and carboplatin work to deform DNA structure to prevent DNA replication, inducing mutations. However, for BRCA-mutated ovarian cancer cells, they cannot use the regular pathway to repair DNA strands. As a result, cancer cells depend on NHEJ as an alternate pathway to repair double-strand breaks, making it resistant to platinum agents. However, the use of poly-ADP ribose polymerase, also known as PARP inhibitors, which is a type of chemotherapy, results in the unreliability of the NHEJ pathway, causing more mutations that cannot be repaired. Even still, PARP inhibitors can also develop a second mutation by using another DNA repair pathway, microhomology-mediated end joining, or MMEJ, which results in the overriding of PARP inhibition by cancer cells, preventing apoptosis for ovarian cancer cells. The cells use various alternate pathways to bypass ones that are inhibited due to treatment, which then results in chemoresistance. The next mechanism of chemoresistance is drug efflux. Efflux pumps moderate the amount of medication in ovarian cancer cells by pumping chemotherapies out of the cancer cell as a defense mechanism. For example, ATP7A and ATP7B are genes that produce proteins that work to efflux platinum, making the cell incapable of using platinum therapy to get treated due to its cisplatin resistance. The cisplatin, which is a chemotherapeutic agent, it's transported through vesicles using ATP to get it across the membrane. Another example of efflux pumps are ABC transporters. So the rate at which the cell is able to pump out the medication exceeds the rate at which the medication enters the cell. As multiple proteins work to moderate chemotherapy concentration, the medication is pumped out of the cell, rendering treatment ineffective. The next mechanism is autophagy. Autophagy recycles remnants of chemotherapy-affected cancer cells to synthesize other cellular structures in other cells. It allows damaged cell organelles to provide energy for the cell through converting the organelles into fatty acids and amino acids through autophagolysosomes, which are made of lysosomes and autophagosomes. However, due to the protective nature of the autophagy process for the cell, it has adverse effects on individuals with cancer. The proteins that are in dying cells are recycled into amino acids and fatty acids in adjacent cells. The energy that is generated through the activation of amino acids allows for cancer cells to continue to rapidly divide and grow. This was demonstrated in a study where they decreased the presence of ATG5 and ATG7, which are proteins directly involved in autophagy. The decrease of the proteins meant that there were decreased amounts of autophagy, which had actually resulted in higher levels of apoptosis, suggesting that the cancer cell is dependent on autophagic processes in order to survive. While chemotherapy is administered in an effort to induce apoptosis of cancer cells, autophagy is used as a defense mechanism to avoid it instead. So despite these main mechanisms of resistance, doctors have developed various treatment methods to combat chemoresistance. The first one being combination therapy. In order to combat chemoresistance of first-line therapies, many doctors try multiple therapies in combination. So the drugs have a different mechanism of action while having improved efficacy. Furthermore, the reduction in dosage to accommodate multiple drugs can actually help alleviate the severity of the side effects of chemotherapy on patients. The drugs work within different types of interactions, synergism and additivism. For a synergistic effect, the two drugs work in tandem to produce a stronger effect than if they were to be administered separately. On the other hand, additive effects are when the individual effects of both drugs are exerted simultaneously. One example of effective combination results are olaparib when administered with carboplatin and cisplatin for platinum-sensitive ovarian cancer patients. Another example is the carboplatin-paclitaxel combination, which results in less toxicity to the nervous system and gastrointestinal tract. The use of combination therapy allows for treatment of cancer despite chemoresistance while also reducing dosage, resulting in a fewer amount of harmful side effects. The next treatment is drug holidays. So this means this is when therapy is continued after taking a break from chemotherapy administration and this is called drug holidays. It makes it possible for cancer cells to regain sensitivity to the drug. As a result, for some patients, they do not have to switch to a second-line therapy and can continue their initial therapy following a break that's determined by a medical professional. This allows doctors to also test the efficiency of the drugs for the patient as opposed to switching to different medications. Now, while treatment is important, another consideration is preventing chemoresistance. When cancer becomes chemoresistant, it often results in a relapse. As a result, if doctors are able to identify continued growth of cancer or its return, they can use this information to assess for altered treatment early on. There are two main methods. One is biomarkers, which are macromolecules that help indicate any changes in biological processes. These can help doctors track the resistance in cancer cells and help predict responses to specific chemotherapies through gene expression of cancer cells. By using this data, medical professionals are able to determine a course of action for different patients. Combination therapy can use fewer cycles to result in a more effective treatment, decreasing the likelihood of developing chemoresistance. Prevention can help reduce the chances of relapse in patients. Now, what is the takeaway? Chemoresistance is the cause of more than 90% of treatment failure for metastatic cancer patients. Although cancer treatment and care has drastically improved over the years, chemoresistance continues to be a significant hurdle within treatment for the increasing number of cancer patients. Addressing chemoresistance is crucial for improving efficacy of first-line treatments. This will help to reduce rates of cancer relapse and positively impact patient survival. Learning about different mechanisms of resistance will allow doctors to be able to identify the best treatment on a patient-to-patient basis. Most importantly, while an understanding of these mechanisms can give insight for clinical implications, it also demonstrates that normal functions of a cell that are typically used to ensure survival are harmful in the context of a cancer patient. As a result, most therapies are developed to bypass the regular functions of a cell. The various types of treatment approaches show that whether doctors change drug administration schedules, giving their patients a drug holiday, or use cisplatin and carboplatin pairs, therapy depends on the patient and not just the disease. However, although treatment is important to help people who are currently suffering from chemoresistance with their cancer, it's important to evaluate how we can eliminate or reduce this problem in the future. It's crucial to implement forward thinking in the domain of public health in order to create prevention strategies. Through studying chemoresistance and its effects on varying patients, doctors will be able to predict the effects of therapies and even prevent chemoresistance to lower ovarian cancer mortality rates. Chemoresistance shows that the prospect of treatment and clinical implications has been altered towards a more patient-to-patient approach. Despite the creation of numerous different treatments, the same approach will not be feasible for all patients because each patient has a unique amalgamation of circumstances and predispositions. Medicine embraces the individuality of patients and the diversity of biology, a science that is not always a standard algorithm with identical outcomes. Thank you.
Thank you very much. Let's see, looking over questions here. So yeah, an initial question here. What of the mechanisms of chemo resistance that you mentioned, I'm curious, which ones seem to be the most important or most dominant mechanisms for ovarian cancer specifically?
So I think this also depends. So chemo resistance is prevalent in all different types of cancers, but this really depends on the type of treatment that you're using. So some treatments, like for example, DNA repair, is more overarching and it kind of affects multiple different treatments. So for example, as I mentioned, the more types of medications you use, the more different pathways the cell will use to try and escape the medications almost. So it's really dependent on the treatment process that you're using in terms of which mechanism is most common. And like again, so drug efflux and autophagy are also different mechanisms that are used against these treatments, but DNA repair does end up being like a more overarching experience of chemo resistance.
Wonderful. And actually, I'm curious from like a personal standpoint, you mentioned at the beginning that you're interested in medicine. Are you interested specifically in oncology or yeah, curious a little bit about how this research pairs with your broader kind of career interests?
Yeah, so I think oncology is definitely a big interest of mine just because like even today if we look in society, cancer is definitely like there's a lot of prevalence. So I think in terms of medicine, oncology is definitely interesting for me. But I think this topic specifically was even more interesting and intriguing because of the fact that it made me realize that although cancer is an issue, it's not the only hurdle when you're going through cancer treatment. So, whereas, yes, you have the symptoms of cancer and you have the symptoms of chemotherapies, chemo resistance also acts as another hurdle. So, I think just really reading about this and understanding that doctors and medical professionals are really looking at all these different types of hurdles while looking at treatment implications, I think was super interesting for me.
Definitely. Interesting to hear how some of the mechanisms that are actually like supposed to be working in healthy cells actually work against for in cancer treatment. So very
Mhm.
interesting presentation. Thank you so much.